Auzubillahiminash-shaytanir-rajim. Bismillahir-rahmanir-rahim. My name is Dr. Muhammad Azhar, Assistant Professor, Department of Genomics and Bioinformatics. Today, my topic of presentation is innovations and annotations in discovering the functional role of and impact of small non-coding and coding genomic elements in the gene regulation. So today we will describe briefly the functional impact of small non-coding and coding genomic elements and their innovations in that field and in gene regulation. As we know that we are familiar with the central dogma that genes are transcripted and translated from the DNA elements into the protein. So it's not a simple process as we know. Currently, the simple central dogma, which we previously know that transcription translation, but with the discovery of 3D genomics and latest sequencing technologies, has revealed that the epigenomic level transcription and post-translation modifications are also important in the central dogma and is also considered as a part of central dogma. So briefly, we know that human genome is composed of 3.2 GB as a genome size, and out of which is only 2.5% is coding and 98.5% is non-coding. And out of 98.5% non-coding, the major part is 80 to 90% is untranslated RNAs, whereas other part are like repeat elements, pseudogenes, transposons, and so on. So we will discuss briefly in a sequential manner that how the post-genetic era led to the genomic era, and now what are the latest technological, experimental, and computational annotation and advancements in discovering the hidden genomic and coding and non-coding elements in different organisms, their functional role. So as we know that with the passage of time in 1990s and in the late 90s and mid 90s, in 1865, Mendel presented the classical Mendel inheritance law, and following that there is massive advancements in the classical and modern genetics with the passage of time. And the first genomic sequencing attempt was on the microgenome influenza and the plasmid, which was made on in 1995, and later it has advanced to the genomic whole genome sequencing of complex eukaryotic organisms. So first whole genome mouse genome project was announced in 1996, and in 1997 the whole genome human genome project was launched, so which was draft completed in 2001 and complete genome finished genome was published in 2003. So here is a comparative analysis of gene number and genome size. As we can see that there is not much difference between the eukaryotic organism between the gene number and the genome size. But so the question arises how the genome complexity comes and will be responsible from the gene elements if the gene number is the same and or similar, not the same, but the genome size is different. The answer is the larger the non-coding part in the higher eukaryotics, which is responsible for the gene regulation, which is responsible for the genome complexity in higher eukaryotes. As we can see the timeline of different genome annotation projects in 1990 and 90, starting from Human Genome Project, leading to the International HabMap project, Human Genome Project, ENCODE project in 2013 for functional genome elements, encoding encyclopedia for coding elements. Then later on, so many projects were launched like non-code, encode, phantom, and 1000 genome projects, and more latest projects like GWAS to date, almost with the 200,000 publications participants. So these all efforts are made to basically annotate the coding and non-coding functional elements. ENCODE project, as we know, is the familiar project encyclopedia of DNA coding elements or non-coding elements, which encounts different kind of sequencing technology like SC, DNase, phase-seq, chip-seq, computation prediction, and RT-PCR, RNA-seq. These are all technology responsible and played a functional and important role in the ENCODE project and the discovery of the genomic and non-coding elements of the and their function role in genome regulation. ENCODE project was launched in 2000, as we know, 3, and the project later in 2006 or 9, and it has been reported at least 80% of the genome is biologically is active, yet only small part of human DNA encodes for protein. Previously, it was thought that the 2.5% genomic code which is coding for the protein is active, but here we can see the 80% or more than 80% out of 98.5% is not inactive, is actually active. So it is playing a function role. So its major part is responsible for producing housekeeping genes, different type of RNAs like ribosomal transfer RNAs and other type of small non-coding RNA, long non-coding RNAs, which plays a functional, practically important, critical function role in the gene regulation, like miRNA, pRNA, link RNA are examples. This is the GenCode project. Was started in 2003 and its second phase was published in 2017 to annotate the human and mouse genome. Here we can see the timeline. The different discovery of miRNA was made in 1992, small nuclear RNA in the 90s. Then we can see miRNA was discovered in 2000 and 2001 or 2, and then pRNA was discovered in 2006. So different type of small non-coding and coding RNAs are recently being discovered. And coding small coding element part example is primary example recently discovered is short non-coding RNA proteins, which plays an important role. So these are major classes of small non-coding RNA divided according to the size and function. And here we can see the expression of RNA abundance and expression, which is high for transfer RNA and miRNA as a number and gene activity role. Sequencing method detection, which usually have been accounted for, is usually small RNA-seq, clip-seq, rip-seq, RNA chip, and these are the methods which have been developed to annotate the coding and non-coding elements, short non-coding elements, and coding elements at genomic level. So these are the sequencing technologies and the experimental advancements in according to the timeline and the bioinformatic advancements. Under the line we can see the different databases and software which have been developed for the annotation of the miRNA, pRNA, and above the line arrow we can see the different type of technologies developed to basically discover, isolate, identify the different types of short non-coding RNAs. So these are the briefly description of recent technologies which we described earlier in this day, earlier in the slide. So these are some most cited tools according to the citations which have been used for the annotation of or discovery of miRNA, pRNA. So pRNA are basically small non-coding RNAs which are in size shorter than the miRNA and accounts for about 20 to 29 or 30 base pairs. Its first discovery was made on the mouse genome as the function role in suppressing the transposable elements. But later on, the different roles in different parts of RNA have been discovered in the gene regulation or the biological experiments and modeling. So here are several modifications have been made in these technologies, in classical existing technologies to the advanced existing technologies, which are like clip technology, advancement of R clip and proximity clip, and latest and so on. So RIP-map is a newer approach which has been discovered in 2023 and has been published in Nature Communications, which is used to identify the short coding RNAs like protein complex, which proteins which are which made a complex, which makes a complex with RNA and other coding elements. So these are microproteins. Microproteins are defined as small non-coding RNAs, which are coding for protein less than 100 amino acids, discovered and coined in MPs in 2013. Methods which have been used to discover are a traditional method, mass spectrometer. Recently, our lab where I did my postdoc, they developed a experiment method equivalent particles to identify the and nano the short coding proteins, which is which gives which gives better results than the traditional mass spectrometry method. So function role of protein are different function roles. Microproteins, their still function roles are not identified, and a large research and experimental area is vacant. So we can scientists can develop and work on this to annotate the functional role of in different biological pathways, the functional role of the short coding RNAs. Here are proteomic analysis. These are the traditional methods: shotgun proteomics, Western blotting, amino precipitation, nuclear magnetic resonance, and these are the traditional methods. But still, we lack the modified and advanced experimental methods to identify, isolate the protein, short coding proteins and their function role, and also we lack the bioinformatic tools and function annotation tools for the prediction. Conclusion: in summary, both non-coding RNA like mRNA, RNAs, and as small coding RNAs like proteins, along with the microproteins, lack sufficient experimental and bioinformatic tools for the effective functional characterization. So improving these resources is essential for a deeper understanding of their roles and mechanism. So here is a large scope and vacant area where scientists can work and develop tools, experimental methods, and work on to explore the functional impact of these short coding and non-coding elements in the genome regulation. So this is all from me. Thank you. Any questions?